the WP1066 was not only very effective in an aggressive GBM mouse model, but when combined with standard radiation treatment, it actually created an immune memory that protected the animals from tumor growth when they were re-challenged with more human tumor implants.
Welcome back for another Virtual Investor What This Means segment. My name is Janine Thomas. I am CEO of JTCIR, and I will be the moderator for the segment. Today, we are featuring Moleculin Biotech, and I am very pleased to be joined by Walter Klemp, Chairman and Chief Executive Officer of the company. Welcome back, Wally.
Glad to be here, Janine. Thanks.
So Wally will go into details on the company's recent announcement regarding the first patient treated in its investigator-initiated phase 2 clinical trial of its STAT3 inhibitor, WP1066, for the treatment of glioblastoma. And before we get started, I want to inform our audience that Moleculin Bio is listed on NASDAQ and trades under the ticker MBRX. And during today's discussion, the company will be making forward-looking statements, and actual results could differ materially from these forward-looking statements. Some of the factors that could cause actual results to differ materially from these contemplated by such forward-looking statements are discussed in the periodic reports Moleculin files with the Securities and Exchange Commission. These documents are available in the Investors section of the company's website and on the Securities and Exchange Commission website, and we encourage you to review these documents carefully. Wally, congratulations to you and the Northwestern University's team on kicking off this investigator-initiated phase 2 study evaluating WP1066 for the treatment of glioblastoma, or GBM. So to start, can you provide an overview of WP1066?
Sure, Janine. So WP1066 is considered a small molecule, and it's designed to target a specific cell signaling pathway that involves a protein called STAT3. Now, STAT3 has long been considered really a holy grail target in oncology because it's considered a master regulator of tumor development. Unfortunately, though, STAT3 has also been considered an undruggable target since researchers around the world have been unsuccessful at creating a drug that's capable of targeting STAT3 without prohibitive side effects. Now, we believe WP1066 may have solved this problem. It's a potent inhibitor of the activated form of STAT3, and yet it's very well tolerated in animals and in humans. And in fact, it's so promising that it has over 50 peer-reviewed articles written about it. And it's been studied by numerous research teams around the world in a range of tumor models.
Thank you. So do you have any data with WP1066 that gives you confidence in its ability to be used for GBM?
We really do. There's quite a bit of data to support the approach against GBM. Now, in the process of demonstrating that the drug is safe in humans, and that was in a phase 1 trial, the oral delivery of WP1066 managed to demonstrate actually a clinical response in 2 children with DIPG. That's a particularly difficult form and deadly form of brain cancer. Now, in addition to all this, Dr. Amy Heimberger, now that's one of the co-PIs in this latest clinical trial, she published a groundbreaking study that showed that WP1066 was not only very effective in an aggressive GBM mouse model, but when combined with standard radiation treatment, it actually created an immune memory that protected the animals from tumor growth when they were re-challenged with more human tumor implants.
Very helpful. Can you speak to your relationship with Northwestern University and what you believe attracted them to wanting to initiate this phase 2 study?
First of all, we're very proud of this collaboration. I mean, Northwestern is a highly respected center for oncology. And in this case, the NIH is actually providing some of the funding for this study. You just don't see this kind of high-end institutional support unless the science is truly compelling and promising. And of course, the need for anything that will move the needle in GBM is extremely high. So the fact that we've already seen some clinical effectiveness with WP1066 in phase 1 combined with the stronger activities that you see when you combine with radiation, that's really created some momentum here.
Okay, so turning to the study, can you provide an overview of the study design and the future milestones for the trial?
Sure. So in general, this is a phase 2. It's an open label multi-arm study of radiation therapy in combination with WP1066. And this is in newly diagnosed glioblastoma patients. Now, the primary endpoint is progression-free survival, or PFS. As we've announced, they've already started recruiting into what we call the lead-in phase, where the first 6 patients will be evaluated for safety and tolerability before they move on with the rest of the trial. Now, all the subjects in this trial will receive WP1066 and radiation post-surgery for tumor removal. What makes this a multi-arm trial is that cohort 1 will be subjects whose tumor was completely removed in that surgery, whereas cohort 2 will be subject whose tumor was not fully removed, usually because it can't be fully removed. That second cohort provides an opportunity to assess the impact of WP1066 in what we call the tumor microenvironment.
Wally, what does success look like for this study?
Well, the biggest potential for a win here, of course, is to see PFS go beyond what's normally expected in glioblastoma patients, which is sadly all too soon. Beyond that, however, what we learned from the study of the tumor microenvironment could provide the whole industry with exciting insights into the potential for STAT3 inhibitors to provide an entirely new way to address GBM, and by the way, a lot of other hard-to-treat cancers.
Okay, so we are here for a What This Means segment, so we'll get right to it. So while your primary focus remains on Annamycin and your lead program in AML, for which you are preparing to launch your pivotal Miracle study, what could success in the WP1066 GBM study mean for Moleculin and its stakeholders?
Janine, you know how excited we are about Annamycin. It's now in phase 3, and it's in a space where we've seen exits in the billions of dollars. That said, I've often explained that WP1066 could be even bigger than Annamycin in the long run. And I say that because STAT3 is considered a master regulator of cancer, and as such is a universal target in which Big Pharma has a very strong interest. Success here would open up an entirely new mechanism of action in oncology, and it's likely to attract a lot of attention from Big Pharma. We believe if we can show sufficient activity in this new clinical trial, it could attract early development partners, and that can represent non-dilutive financing that can have a very meaningful impact on our market capitalization and very beneficial for our shareholders.
With that, this concludes our What This Means segment with Moleculin Biotech. I'd like to extend a huge thank you to Wally Klemp for joining us today. I'd also like to thank our viewers for watching this segment. As a reminder, Moleculin trades on NASDAQ under the ticker MBRX. And if you like what you saw today, I encourage you to visit moleculin.com for more information on the company, to sign up to follow the company to receive their alerts, as well as follow their social channels to stay current on the latest information. You can also visit virtualinvestorco.com for a replay of today's segment, as well as our latest segments and events calendar. I want to thank everyone and wish you a great rest of your day.